When I was 3 years old, I was obsessed with touching a hot pan. When I was finally successful at completing this very odd task, I yelped and pulled my hand away. Little did I know this was the first time I saw mechanical signals dictate movement. Our cells, a lot like my 3-year-old self, get a lot of information from the physical inputs around them. I am studying how those inputs tell our cells to move. Beyond wanting to understand my reaction all those years ago, understanding how our cells know where and when to move can let us better treat cancer. 80% of patients who have tumor cells spread from one area to another will die within 5 years. This process, called metastasis, occurs when tumor cells move when they aren't even supposed to. If we have a better understanding of where those signals are coming from, we might be able to prevent metastasis in the first place. Our cells move a lot like a hamster in a ball. There's this protein in teal called actin. It pushes up against the cell membrane, the dark pink. Actin headbutts the cell membrane to push the entire cell forward. But instead of taking a step like a hamster, it grows longer in the direction it wants to push the cell. In the lab, there isn't really a great way to mimic the mechanical signals imparted by the cell membrane. But that's where I come in. DNA helices are formed when the letters of 2 DNA strands, called the sequence, come together and hold on to each other. You may have learned that A goes with T and C pairs with G, but it's actually the number of hydrogen bonds each of those letters can make that tells them how to pair up. I leverage this fact. By changing the letters in the sequence, I can alter how many hydrogen bonds a strand is making and thus how strongly they hold on to each other. I attach the blue strand onto actin and the matching red strand to a piece of glass. At first, actin grows normally, but once it gets long enough for the 2 strands to come together, boom, a helix is formed, and actin is no longer able to grow normally. Actin is stuck there until it breaks the bonds between the 2 pieces of DNA. Using a microscope, I then watch how fast actin grows as these forces are applied. And I found that 43 piconewtons can stop actin from growing indefinitely. This means if we applied the equivalent force onto a tumor, we might be able to stop metastasis from occurring. While all this work is clearly in its infancy, it's clear that my DNA force probe can be used to quantitatively study the forces that dictate cellular movement. In fact, this tool can be used throughout the cell to study how cytoskeletal dynamics change under force. But perhaps most importantly, I've learned not to touch a hot pan. Thank you.